We are in biomedical instrumentation course. In this course, today we're going to discuss origin of biopotential and its propagation. Yes, what is potential? What is biopotential? Generally, the voltage which will be measured, generated, and it will be measured is called potential. So the difference in the flow of current should be measured at 2 points. Here we are dealing with bioelectric potential or biopotential. Here in the cells, the potential will be generated. It should be recorded. It should be observed by using electrodes. Why we are measuring this potential and measuring these electrodes? For accessing or for measuring the brain activity, heart activity, or muscle activity, and so on. So the bioelectric potential or the ionic voltage which produced to the electrochemical activity of cells associated with the nerve conduction, brain activity, heartbeat, muscle, and so on. So here I told that it's associated with cells. Then what is a cell? Actually, the basic living unit of the body is the cell. To understand the function of organs and other structure of the body, it is essential to know about the basic organization of the cells and function of its component. So in our body, each organ is aggregate of many different cells, which may held together by intracellular supporting structures. So each cell which perform one function, some functions. So in our body, we may have more than 100 trillion cells. From these 100 trillion cells, 25 trillion cells are RBCs, which is called red blood cells. It helps for transporting the oxygen from lungs to the other tissues. So, like that, each cells have the ability to reproduce a new cell. Because for the particular cells, when the cells may be destroyed, so it cannot meet the appropriate number. After destroying some cells, automatically cells also create another cells to replenish and it do this activity keep on. That is the magic thing which will happen in our body. So from these cells, we may take some potentials. That potential is called biopotential or bioelectric potential. Next, the living tissues. The living tissue are considered as the power station for generating the multiple electrical signals within the two internal sources. The two internal sources is called muscles and nerves. Two internal sources which is called muscles and nerves. So this muscular contraction which is associated with the migration of ions. The migration of ions means we have anion and cation. So these migration of ions which generates the potential differences. These potential difference of potentials is called action potential and bio resting potential. So the concentration ions mostly in the cells are called resting potential. Okay. Before that, we will deal with bioelectric potential, which can be measured or generated at the cellular level. The source of the potential is always in ionic nature. So our human cell vary from 1 micron to 100 micron diameter. Its length is about 1 millimeter to 1 meter. The thickness of this membrane is about 0.01 micron. Each cell consists of an ionic conductor, which will be separated from the outer environment by a semi-permeable membrane, which act as the selective ionic filter to the ions. So in our body fluids, we surround the cells of the body are ionic and provides the conducting medium for electric potentials. Now we may think about the excitable cells. What is excited? The cells are excited. Then it is called excitable cells. How the cells are excited based on our muscles and nerve cells, which are stimulated by creating a tiny electric current. So our muscles and nerves cells are stimulated by a small electric current, which is called excitable cells. The membrane of the excitable cells, which permits the entry of potassium, which is called K plus, and chlorine, which is called Cl minus, and also it should block sodium Na plus ions. So, due to the difference in the permeability of different ions, the concentration of sodium ions inside the cell becomes much lower than the outside of the cell. So, generally, the sodium ions are positive. The outside cell is more positive than inside. Similarly, the concentration of potassium and chloride ion is more inside than the outside. Thus, the charge balance is not achieved. So, we need equilibrium to reach with this potential difference across any membrane that is called positive or negative. So, we have the movement of positive and negative ions in the cells. Coming to the biopotential, there are two type of potentials are there. One is acting action potential, another one is resting potential. What is action potential? What is resting potential? When a cell has slightly positive on the inside due to the imbalance of potassium ions, this positive potential of the cell membrane, which may be excited up to 200 millivolt. This type of potential is called action potential. Similarly, when the cell which is in the resting state, which means a cell has a negative charge along the inner surface and positive charge along the outer surface, this membrane potential is called resting potential of the cell. So, similarly, we may have cells which have positive and negative. Generally, in the normal resting state, the cell is interior, it's negative with respect to the outside. Whenever the cell got fired, the outside of the cell becomes negative with respect to the interior. Similarly, after a short time, the cell again becomes to the normal state, which again, with respect to the outside, the discharging and recharging of the cell can take place, which is called depolarization and repolarization. The charging, recharging, discharging. So the discharge is called depolarization, recharging is called repolarization. So it may happen based on the interest will be generated by the cells. So a group of cell which is called tissue. அதுதான் டிஷ்யூன்னு சொல்லுவோம். அந்த டிஷ்யூல நர்வ் செல்ஸ், கிளாஸ் செல்ஸ், ஒரு நர்வஸ் சிஸ்டமே நர்வ் செல்ஸ் தான் மேடப் பண்ணுவாங்க. ஸோ பேஸ்ட் ஆன் த boundary axons, the nerve cells with the dendrites and fiber, which is called neuron, that is fire neuron. So that from that neuron, we may get the information. It should be transmitted through axon by a small impulses, which is called a constant amplitude. From that, this energy, these cells may get charge and it should be connected with that. இதுதான் நம்ம வந்து பொட்டன்ஷியல் அப்படின்னு சொல்றோம். Let us see the diagram. What is it? Initially, it is a resting state, which means inside we have negative potential, outside we have positive potential, which is a polarized state. After that, we may get excited state. In the excited state, we may have a part of the cells becomes positive and part of the cells become negative. So when the cell is in the resting state, which has a negative along the inner surface and positive along the upper surface, so this membrane potential is called resting potential of the cell. So it is maintained and some kind of disturbances, it should upset the equilibrium. A cell which is in the resting state is called polarized state. Next, depolarization, repolarization. Same cell. When the cell is excited, we come in this area. When cell is excited or stimulated, the outer becomes negative. The outer becomes negative. With respect to the inner becomes positive. Similarly, in an excited, inner becomes negative, outer becomes positive. So this process which is called depolarization, the process perida depolarization, which is called discharge of the cells. So these cell have the potential changes with up to plus 20 millivolt. Okay, after depolarization, again it becomes a Lie polarization mastered. So after the depolarization, the cell is totally unpolarized, which is called depolarized, opposite to the polarized. Inside we have the positive, outside we have the negative. This state becomes a unpolarized state. Again, the repolarization takes place. It is a short time later when the cell again regain the normal state, that is, it becomes positive inside with respect to the outer surface. So the cell has slightly positive potential on the inside due to the imbalance of the potassium. Due to the imbalance of the potassium ions. Again, this positive potential of the cell membrane during the excitation, the action potential is which is about at 20 millivolt. After completing total repolarization, the cell becomes in the resting states. So the cell becomes in the resting state. Hope you may understand the propagation. It is very important. Polarized, stimulated, depolarized, repolarized, and become a polarized state. So when a cell is in the resting phase is called polarized. The measurement of the membrane potential which is made inside a cell, which may given as negative, which is nothing but minus 60 millivolt to minus 100 millivolt. It is very important. So the resting potential of the cell have a negative value, which is range about minus 60 millivolt to minus 100 millivolt. Next, what I told, excited, stimulated, which are on. So the typical cell potential waveform. So here we have the polarized. This is the polarized cell, which is called resting potential minus 60 plus 20, which is called the duration minimum width of the current stimulus for the action potential generation. This period is called stimulus period. So plus 22 minus 60. This state is called depolarization state, which is called discharging state. This is called repolarization, which is called recharging state. So the potential waveform shows the depolarization and the repolarization of the cells. Continue with that, we may have a perfectly polarized or perfectly unpolarized. The electrodes in which no transfer of charge occurs the metal electrolyte interface, which is called perfectly polarized. Which is called perfectly polarized. Similarly, electrodes which are exchange of charge occurs due to the metal electrolyte interface, which is called perfectly non-polarized. Which is called perfectly non-polarized. Okay. Next, what is absolute refractory period? What is relative refractory period? when the time duration of the cell non-response to the further stimuli or any stimuli, then this period is called absolute refractory period. It is about 1 millisecond in our nerve cells. So total absolute refractory period, which is not non-response to the any stimuli, that particular time duration, which is called absolute refraction period. What about the relative refractory period? It may generate the absolute refractory period. There occurs of the relative refractory period during which any other action potential can be taken. So it also about several milliseconds in nerve cells. So which is called relative one compared with the another, right? Next, perfectly and non-perfectly. And all or nothing law. Actually, regardless of method of excitation of cells or intensity of the stimulus, our action potential, which is always same for any cell, any given cell. So our action potential is same for any given cell. Even our method of excitation cells or intensity of the stimulus, which is changed, which is called all or nothing law. Fine. One of the important to mark. Next, types of biopotentials. Types of biopotentials. So we are dealing with bioelectric potential. How our bioelectric potential related? If you want to measure the heart, the methodology of measuring the bioelectric potential is electrocardiogram, which is called ECG. For measuring the brain, we may call electroencephalogram, which is called EEG. Third one, muscles. If you want to measure the muscles, the name is electromyogram. While eye, the retina of the eye, which is called electroretinogram, ERG. Then eye, cornea and retina, which is called electrooculogram. So these are the some bioelectric potentials from our human body. We may measure the potentials. This methodology name is called ECG, EAG, EMG, ERG, and EMG. See, we have to think. Uh, what is next one is half cell potential or electrode potential. When voltage develop at electrode and electrolyte interface. See, we are dealing with electrode and electrolyte. Electrolyte, which is a changes of potassium and ions. This will be deal with electrode. So these type of interface so that the voltage will be developed. This design is called electrode potential or half cell potential. So in the case we have a metal solution in the surface and they have another electrode potential which will be result from the different rate, which is opposite process. Let us have a ions from the metal and into the solution. So when metal electrode comes into contact with an electrolyte, there is a tendency of the electrode to discharge ions into the solution so that ions in the electrolyte to become combined with the electrode. The final result may have a creation of charge. This arrangement, this creation, this arrangement, which is called electrical double layer, இதுதான் நம்ம என்ன சொல்லுவோம், there is a no net transfer of charge occurs at the metal electrolyte interface is called perfectly polarized electrode. The same thing, a hinder exchange of charge across the metal electrolyte interface, which is called perfectly non-polarized. So these are the thing which will be occurred in this potential option. So latency, sodium pump, please note down latency, which is the elapsed time between the stimulating pulse and the muscle's action potential. This time period is called latency. And what is the sodium pump? Actually, it is one of the active process by which sodium ions are quickly transported to the outside of the cell, and the cell again becomes polarized and assume its resting. This operation of this pump is linked with the influx of potassium into the cell, as if a cyclic process involving an exchange of sodium for the potassium existed. Hope These are the things which will be coming under the origin of biopotential. Thank you for listening. We will go to the next topic, the next video, which is called types of electrodes. Thank you, Regine. Please stop this recording.